Hello, everybody, and welcome to today's Revive webinar by the Global Antibiotic Research and Development Partnership, GARD-P. Today's session is about influence of biological sex in infectious diseases. A little background on our program. So Revive is an online space to connect and support the antimicrobial discovery, research and development community, and these webinars are part of our educational activities. All our webinars are recorded and are available on our website under revive.gardp.org/webinars. And also today's video will be added there within approximately 1 week. I invite you to go to the Revive website also to look for other content. For example, we have an antimicrobial viewpoint series with articles about various topics related to AMR, and an antimicrobial encyclopedia, which offers short definitions and explanations for key terms in the field. And each term also is complemented with further reading materials, and some terms are even explained by expert videos. For today's session, apologies, our moderator will give a short introduction on the topic, and then we have 2 presentations. And after the final presentation, we will host a Q&A session. But you can submit your questions at any time during the webinar by adding them in the questions window in the control panel. I would just ask you to please include the name of the person your question is addressed to. So now I'm happy to welcome today's panel. We have Molly Ingersoll from the Institut Cochin and Institut Pasteur in France, and Sabra Klein from Johns Hopkins Bloomberg School of Public Health in the US. And our moderator is Neeloffer Mookherjee from the University of Manitoba. Neeloffer is a professor at the University of Manitoba, where she leads an internationally recognized research program on chronic inflammatory diseases with a focus on immunity-related functions of molecules known as host defense or antimicrobial peptides. As the Canadian Institute of Health Research Sex and Gender Science Chair in Respiratory Health, she integrates sex and gender-based analysis in her research program and examines sex as a biological variable in the regulation of inflammation in the lungs. Neeloffer is a strong advocate for women in science and is the current chair of WISDOM, that stands for Women in Science Development, Outreach and Mentorship initiative at the Max Rady Faculty of Health Sciences at the University of Manitoba. Neeloffer, welcome to this session. Please switch on your webcam now, and I will give you control over.
Thanks, Astrid.
Your slides.
Thank you. Thank you for the kind introduction. Welcome, everybody. I'm going to be introducing the topic very briefly and introducing yet speakers for today's webinar. The topic is influence of biological sex in infectious disease. And I would like to start by acknowledging or addressing the difference between sex and gender. As some of you have seen in the media, there has been some interest in accounting for sex and gender in biomedical research. And there are some incredibly good review articles that talk about how accounting for sex and gender just makes for better science. And I especially like this quote of the scientific director of Canadian Institutes of Health Research, Institute of Gender and Health, Dr. Kara Tannenbaum, who said very poignantly, women are not just small men. So first, let's begin with what is sex and gender and the difference between sex and gender. So sex is the biological attributes of humans, animals, which includes biological components such as chromosomal makeup, gene expression, hormones, and anatomy. So biological sex or sex would include males, females, intersex. Whereas gender is socially constructed roles of behavior, expressions, and gender identities of girls, women, boys, men, and gender diverse people. For those of you who may be interested in divulging a bit more into the basics of sex and gender, there are incredibly good instructional modules hosted by the Canadian Institute of Health Research, or CIHR. And here is the URL. It's open to all and talks about sex and gender training for both clinical and non-clinical scientists. So together with sex and gender, we have many other variables, and the intersectionality of these variables have an impact on biology. So the intersectionality of sex and gender would include age, race, and other gendered identity and behavior related variables. But for today's topic, we are focusing on biological sex. That is the biological attributes of humans, animals, which includes, again, for your reminder, chromosomal makeup, sex, steroids, and gene expression and anatomy. So why, as biomedical scientists, should we look into sex and gender? As it is evident from research in the last few years that biological sex as well as gender constructs have direct effect on disease pathophysiology, severity, manifestation of disease, as well as response to treatment. Specifically with biological sex, as you might well understand now, that chromosomes as well as sex hormones have direct impact on disease pathophysiology. Gender as well has an effect on biological sex, so gene expression, protein expression, and we are not going to be looking into this very complex setup for today's talk. For those of you who may be interested, this particular figure has been taken from the Lancet review article in 2020, talking about sex and gender as modifiers of health and disease. It is thus not surprising that sex as well as gender, in this case sex, has a bias in autoimmune disease, reproductive cancers, and as well as infectious disease. For today's webinar, we are going to be focusing on the influence of sex in infectious disease. With that, I would like to welcome our first speaker, Dr. Sabra Klein. Dr. Sabra Klein is a professor in molecular microbiology and immunology at the Department of Medicine in Johns Hopkins School of Medicine and International Health at Johns Hopkins School of Public Health. Dr. Sabra Klein is a world expert in sex and gender differences in immune responses, susceptibility to infections and vaccines, with examination of sex-specific effects of aging in humans and animal models. Dr. Klein has over 175 peer-reviewed publications, several authored book chapters, has edited numerous books on broad topics of sex differences in response to infections and immunity. To be noted, during the 2009 influenza pandemic, Dr. Klein was commissioned by the WHO to evaluate and publish a report on the impact of sex, gender, and pregnancy on the outcome of influenza virus infections. During the current COVID-19 pandemic, Dr. Klein has written many reports and commentaries in several journals and has been interviewed by major news media outlets about sex differences in immunity, disease outcomes, and response to vaccines. I am delighted to welcome Dr. Sabra Klein. Dr. Klein, the stage is yours.
Thank you so much, and what a wonderful introduction to our topic today. And I'm going to be very focused on respiratory diseases. I'm not certain I have control. Oh, I do. So before we launch into discussing respiratory diseases, I thought I might begin by discussing sex differences in viral diseases and what we understand from the literature, what we are still learning from the ongoing pandemic as COVID-19 moves into the endemic phase. And what I've done here is I've graphed immune responses, which can range from weak to strong, relative to the damage that infections can cause, which can range from a low degree of tissue damage to a high degree of tissue damage. And many of us in this field will be very familiar with thinking about a disease response curve, which is illustrated in black. And we can have a high degree of tissue damage and disease if we have a weak immune response and are unable to control a virus. And if we follow that curve around, we can also have a high degree of tissue damage and disease if our immune response is too strong, and that excessive immunity can contribute to what we often term immunopathology, or where the excessiveness of the immune response is contributing to the tissue damage. And what I've done here is I've drawn a stippled line in the middle, and I've put males on one side and females on the other. And as was illustrated in the introductory remarks, we do have infectious diseases for which we see a male bias, meaning that males suffer a worse outcome. And you'll note that SARS-CoV-2 is on this list. And then alternatively, we absolutely, though not as plentiful, we do have infectious diseases for which we note that females show worse disease outcomes. And what I hope to do today is provide you some evidence, and we'll see if all of my data actually support these hypotheses. And I think what I hope to do today is illustrate that the extent of these differences between males and females are often dependent on age. So let's look at some epidemiological data that support this notion that there can be an intersection between sex and age. Here we have data from Australia, post the 2009 H1N1 pandemic, so from 2010 to 2015. And what I loved about these data, they broke down their data, and these are all clinical data referring to notification rates. And so as many of you on this webinar know, influenza is notoriously underreported, in part because it causes an acute infection. It causes this kind of nondescript influenza-like illness. Most of us stay home, we don't seek care. But if you are sick enough to seek care and be confirmed to have influenza, I think we could use that as a marker of severity of disease. So that's what this is referring to when it says notification. Individuals who are sick enough to go to the hospital in Australia to be confirmed to have influenza. And so that's what's here on the y-axis. On the x-axis are ages ranging from very young to very old. And then on our secondary y-axis, we have the relative ratio of females relative to males being hospitalized with influenza. And on this secondary Y, a 1 indicates no difference between males and females. So for simplicity, if we use this, and these are these green symbols, we find that there are fewer females, in other words, more males being hospitalized with flu at very young ages, as well as very old ages. And in the middle is where we see more females being hospitalized relative to males. Pregnancy is absolutely an important factor, and maybe we'll convince this organization to do a webinar on pregnancy. I'm not going to talk about pregnancy any further, but that is a sex-specific factor that does contribute to worse outcomes from many infectious diseases. But it turns out that just being female, so even among non-pregnant individuals, we do find that females of reproductive ages in through menopause are more likely to be hospitalized with influenza. We've seen this for seasonal epidemics, we've seen this for pandemics, and we can model this in the laboratory. So working with C57 black 6 mice, females in purple, males in green, when we infect intranasally these mice, and these are young adults, with 2009 H1N1, females get sicker. They lose more of their body mass as compared with males. When we look at virus replication in the lungs, the primary site of virus replication, we do not see differences between males and females, either during peak virus replication or as clearance occurs or when clearance has been completed, with LOD referring to the limit of detection. We can look at the function of the lungs as maybe a better indicator than body mass of how sick these animals get. And if we look here, 14 days after infection, virus has been cleared, males have returned back even above baseline health, that's that stippled line. Females are still in that recovery phase, and we see this here for the lungs. When we look at the diffusion capacity, DFCO, and that's the diffusion or exchange of oxygen and CO2 across the lung, males are back to baseline health as indicated by these stippled lines, females are not. Histopathologically, when we look at the lungs, we find that females, even out 14 days, this is an acute infection, 14 days after infection, virus is cleared, females still have inflammation, and within that first week after infection, we've reported for a number of years, females have greater induction of cytokines, including TNF and CCL2. Everybody survives. Young adult mice, much like young adult people, we survive influenza infection. It is very rare to have a death. And once these animals survive, what we find is that females go on to have much more robust immunity. They have greater antibody responses, both in their serum as well as in their bronchoalveolar lavage fluid. And then in the lungs, they have more antibody secreting cells than males, more CD4 T cells, greater numbers of CD8 positive T cells, both virus specific, and here we're looking at memory, as well as resident memory T cells. So although females get sick in that primary infection, sicker than males, once they've recovered, they have greater immunity. We also find that females have greater immunity after vaccination. So if we vaccinate young adult male and female C57 black 6 mice intramuscularly with influenza, we find that, and we do this using a prime boost strategy, females have greater IgG as well as neutralizing antibodies. So that's antibody that's going to neutralize or prevent virus from infecting susceptible cells. And when we look at the avidity of that IgG, the avidity is greater for females, the open symbols, as compared with males. And we do this by using a chemical that causes dissociation of the antibody-antigen complex. Not only do females have greater antibody, they have greater antiviral antibody. And so in mice, there are many flavors or isotypes of IgG. And when we look at IgG2C, which is the antiviral isotype of IgG in C57 mice, females have more of this antibody. And a key regulator of class switching, that class switch recombination that occurs in B cells, is toll-like receptor 7 or TLR7, which when we isolate B cells from vaccinated males and females, females have greater expression. You may not know this, but TLR7 is encoded on our X chromosome. And there is substantial data, both in humans as well as in mice, with our data suggesting that TLR7 can actually escape X inactivation and can be found in greater copy number in females as compared with males. Well, working with inbred mice, we can't exactly look at the maternal and paternal expression of TLR7. So we used an epigenetic approach looking at DNA methylation in the promoter of TLR7 and observed reduced DNA methylation in the promoter of TLR7 in vaccinated females as compared with males, suggesting that reduced DNA methylation might be one factor contributing to greater expression of TLR7 and greater activity of antiviral antibody in vaccinated females compared with males. When we take these vaccinated males and females and challenge them with a live drift variant of the 2009 H1N1, we observe when we look in the lungs for replicating virus, females the open symbols, males the closed symbols, females clear virus faster within that first week after infection. And when we look for 2 weeks at markers of disease, females really don't change from their baseline health, that stippled line, whereas you can see males get sick. Everybody survives. It's not to say vaccines don't work in males. This is not to be alarmist. It's just to say they're working better in females. Not only do females have greater antibody responses, but when we look at the short-term producers, the immediate producers of antibodies, such as plasmablasts, we find that vaccinated females in purple, males in green have more plasmablasts, whether we look at the number or frequencies of these cells. And when we look in the draining lymph nodes and look at germinal center B cells, which are going to give rise to both the plasma cells and the memory B cells, females have more of these cells as well. Within these germinal centers, females have greater expression of the gene that encodes for activation-induced cytidine deaminase. And this is important for regulating somatic hypermutations. So when we look at the expression of this gene in germinal center B cells, females have greater expression. And when we actually sequence mutations in the JH4 region from germinal center B cells, females have a greater mutation frequency. So we're seeing more mutated sequences in germinal centers from B cells. And when we look on a per sequence basis, with lighter colors indicating fewer mutations, darker colors indicating more. When you look at the size of this pie, a majority of the sequences from males have no mutations, whereas a majority of the sequences from females have 10 or more mutations. This all changes with age, so this is very age specific. We only see the sex difference in numbers of plasmablasts, numbers of germinal B cells, and in the frequency of mutations, these somatic hypermutations in young adult females. I see that the color disappeared from my slides, but you can see these are aged males, aged females. We lose the sex difference both in the frequencies of these cells as well as in the mutation frequencies. Sex steroids are intimately involved, both in humans and in mice, in regulating these differences. When we look at circulating estradiol concentrations among adult and aged females, adults being 18 to 49 years of age, aged being 65 years and older, as you can see, estradiol concentrations are significantly reduced. When we look in mice, adult female mice being roughly 10 to 12 weeks of age at the time that blood was collected, aged females being roughly 68 to 72 weeks of age at the time that blood was collected, you can see that we can really mirror what we see in humans in our mice. If we look at the association of estradiol concentrations with neutralizing antibody responses, and these both in humans and in mice are against the 2009 H1N1, we see a positive association both among aged females, abbreviated AGF, as well as adult females, abbreviated ADF, in which females with greater circulating concentrations of estradiol have greater neutralizing antibody responses. So we can take this and think about this in a therapeutic way, and maybe it's because of my age, I'm starting to think about estrogen replacement therapy. So if we take our mice and we give either adult or aged animals a placebo pellet, and give other adult and aged female mice an estradiol containing pellet that time releases this hormone, what we find and what I've shown previously, adult females have greater antibody responses post-vaccination as compared with aged females. And when we give females estradiol, this results in responses that are indistinguishable from adult females that have endogenous estradiol. So we were really excited to see that we can improve the neutralizing antibody response in aged females by administering estradiol. When we challenge these animals with a drip variant of virus, these aged females that have estradiol are undistinguishable from young adult females that have either endogenous estradiol or exogenous estradiol, suggesting that estradiol is in fact protective. I'm just going to keep moving to keep us on time and switch gears to talk a bit about SARS-CoV-2. During the first wave, we very quickly recognized that there was a male bias, both in hospitalization as well as in ICU admission and death. When we looked at case fatality rates across diverse countries that were disaggregating their data, broken down by sex, what we observed was that even among countries, well, whether these were countries that had an extreme case fatality rate or those with a less extreme case fatality rate, we were seeing roughly twofold increased rates of death among males as compared with females. Among a handful of countries that actually disaggregated data not just by sex, but also by age, we observed that across diverse ages, I should say across diverse adult ages, males had greater case fatality rates than females, but this is not to ignore the age-associated increase in death rates that were apparent among females at these older ages. At Johns Hopkins, using electronic medical records, we looked within our own data sets and observed that when we looked at, of those who were tested, who was testing positive for SARS-CoV-2, and we observed that a greater proportion of those tested were males among individuals 18 to 49, upward to 65 to 74 years of age. And among those who tested positive for SARS-CoV-2, when we looked at who was sick enough to be admitted into the Johns Hopkins hospitals, a greater proportion of males within these age ranges were admitted. We observed greater inflammatory biomarker responses immediately at the time of admission into the hospital. So within 48 hours of admission into the hospital, among males who were admitted into the hospital as compared with females. And you can see these differences for some, not all of the biomarkers are listed here, but these include ferritin, IL-6, a cytokine, C-reactive protein, a very potent clinical biomarker of inflammation, males blue, females red, and you can see we saw this difference, with this difference actually being most pronounced among the younger ages as compared to the older ages. I'm just going to jump here and tell you how we use biostatistics in order to test hypotheses about mechanisms that could be mediating sex differences in hospital admission in our population. And what we consistently observed was that using biostatistical modeling in which we removed various factors for which we observed sex differences, it was really only the removal of baseline inflammatory laboratory data that eliminated the sex difference in hospital admission and outcome, suggesting to us this was a testable hypothesis. So to test this hypothesis, we go back to the bench, back to our animal models. And so using golden Syrian hamsters, which are a wonderful model in that they are susceptible to clinical isolates of SARS-CoV-2. Neither the animal nor the viruses need to be manipulated. And what we observed when we gave intranasal inoculation with the initial Wuhan strain of SARS-CoV-2, males the closed symbols, females the open symbols, we observed that males got sicker. When we looked at using CT scans of the pulmonary tissue, We observed in an unbiased manner, males the upper, female the lower, we were observing evidence of pneumonia. So we were seeing some of those opacities, those ground glass opacities that are consistent with pneumonia. Creating 3D images of the affected lung relative to the total lung, you can see that there was a greater percentage of affected lung in males as compared with females, despite there being no difference between males and females in the ability to control virus replication. So despite our initial hypothesis that males may be less capable of controlling virus, that was not what we observed. We also observed that both males and females experienced inflammation. So both were having markers of inflammation in their lung tissue, and that's what's shown in panel A. But using the few flow cytometry markers that were available to us, and there were not many for hamsters, so the downside of working with these species are the lack of reagents, males were showing increases in myeloid cells in their lungs, whereas females were showing increases in lymphocytes, suggesting that the immune responses and the induction of immune responses may be different between the sexes. When we looked at antibody responses, consistent with what we've observed for flu, females the open symbols, males the closed symbols, whether we look in blood or whether we look locally in the lungs at something like IgG recognizing SARS-CoV-2, we consistently observe greater antibody responses in females as compared with males. So again, we see these sex differences, and the last point that I want to make with you, as many of you may be doing clinical research, you might be doing epidemiological research, you might even be doing animal work and questioning whether you really need to consider sex. And so I just want to bring your attention to a paper that we published last year using our own human data, which I did not have time to get into today. And so what we did with our own human data from human influenza vaccine studies, as well as from COVID-19 mRNA vaccine studies, if we treat sex as a confounding variable in our clinical research, meaning that we control for sex or gender to control, or if you will, eliminate the impact that this might have on a predictor, in our case age, on an outcome of interest, in this case the antibody response to a vaccine antigen, and we look at what this does over age, what we find is that we see no age-associated difference for either males or females in the response to the influenza vaccine. And for COVID, we see a significant effect of age that's the same degree in both males and females. Now, if we take these exact same data and now treat sex and gender as an effect modifier, as something that might be intersecting or interacting with our predictor, again, age on the outcome, again, vaccine antibody responses, now what we observe for both responses to influenza vaccines and COVID vaccines is that that age-associated decline in antibody responses is only observed among males, not among females. Among females, we see no significant change in the antibody response. So we miss out on that intersectionality, which may better explain targeting. It may even better explain the variability that we see in the therapeutic benefit of our interventions, including vaccines. I'd like to thank everybody who works with me. I have wonderful lab and colleagues. I thank my funding, and I thank you for your time.
Thank you so much, Dr. Klein, for that amazing talk. And for the audience, we will ask the questions at the end of the second speaker's talk. Please write your questions in the question window and indicate which speaker you would like us to ask the question to. With that, I'm delighted to welcome our second speaker, Dr. Molly Ingersoll. Dr. Molly Ingersoll received her PhD in host pathogen interaction from NYU School of Medicine in New York City. Dr. Ingersoll did her postdoctoral training at Washington University School of Medicine, as well as studied innate immunity, the urinary tract infections. She moved back to New York for another postdoctoral fellowship at Mount Sinai Medical Center, investigating monocyte and dendritic cell biology. Then Dr. Ingersoll moved to Paris in 2012 and established her research group at Institut Pasteur. Currently, Dr. Ingersoll has dual affiliation as a research director at Institut Cochin and Institut Pasteur, where Molly and her team investigate mucosal immunity in the bladder in the context of infection and cancer. Welcome, Dr. Ingersoll. The stage is yours now.
Thank you so much, Neeloffer. Thank you. See if I can pull up a pointer here. It's so tiny. All right, great. Thank you so much. Thanks for the invitation and for the very nice introduction. As Neeloffer said, my name is Molly Ingersoll, and you can find me here on Twitter, or you can send me an email. And currently, I'm the head of the mucosal inflammation and immunity group at Institut Cochin, with an affiliation with Institut Pasteur. So in the lab, we're really curious about how mucosal immunity develops specifically in the bladder. We are interested in part because it's an important mucosal site, but it's also a very understudied mucosal site. And the way we do this is we look at the response to diseases such as urinary tract infection, but also bladder cancer. And one of the things that we think is really interesting about these is we can use the studies from one to understand mechanisms that occur in the other. And ultimately what we want to do is we want to identify targets for immunotherapy so that we can ultimately improve patient response in both of these disease contexts. And one of the things that we're really quite interested in is that both of these diseases have really strong sex biases. Bladder cancer is much more common in men, and urinary tract infection is much more common in women. And a lot of the times we get the question, actually, do only women get urinary tract infection? And one of the explanations that we hear frequently for the number of women who get UTI is that infection occurs much less frequently in men because their urethra is much longer. And so we're going to just talk about that for 1 second. So if we consider among patients who are adolescents or adults, this is sort of what the patient population looks like. You can see that it's primarily women, so they're in red. And men here are in blue. So not that many men in this age group get urinary tract infection. But if we step back and we look at sort of the whole population in general, what we see is about 50% of women in this age group are going to get at least one urinary tract infection in their lifetime, whereas about 1% of men will get infection. But if we consider the two ends of life, if you will, if we consider infants, for example, we see that the incidence of urinary tract infection is actually higher in baby boys compared to baby girls. And if we look at the other end, we see that the incidence of UTI actually decreases in women over the age of 50, while it increases quite dramatically in men over the age of 50. And so what I've put down here at the bottom are the percentage of the total population that can expect to be infected at any given time. So you can see this is really prevalent infection. But you also see that the incidence between women and men actually changes over time. And one of the things that I think is really important here, and the reason that I stamp this statement as false, is we do know that, for instance, the urethra doesn't change between adult men and men over the age of 50. And so what that suggests to us is that there are other factors that influence susceptibility to urinary tract infection and probably also our response to urinary tract infection. So one of the first questions we asked is, if this is the case, how are we going to be able to test this? And the reason that this was an important question for us is, how do I put this? It's sort of the reluctance we faced in the literature to address this type of question. And ultimately what we ran into were publications that said intravesical installation. And so this is the act of placing a catheter into the urethra of an animal, for example, to induce an infection. This is only possible in females. It's anatomically impossible in male animals. Other publications state that they use primarily female animals because they can be catheterized, or because males have a longer urethra than females. Doing this catheterization is not possible. So I wouldn't be here today if that was the case. And in fact, it is possible. We had a ambitious student in the lab who looked into the literature, and she found a method to essentially do exactly this. And so what we did is we developed a way to put a catheter into male animals so that we can really ask the question, is there a difference in not so much susceptibility but response to infection between male and female animals? And if you're curious, you can see a video of how we do this down here. So that's published. And so ultimately what we did, so this is the work of Anna Zaklinski-Scharf, who was a student in the lab at the time, and she just asked the question, what happens when we infect male and female mice? And so what we did here, what you're looking at, is the number of infected mice on the y-axis over time. And what she did is she infected equal numbers of male and female mice, and she looked at resolution of infection over time. So essentially what she did is she took urine from these mice and she just looked to see if there was bacteria present or not. And when the bacteria were gone, then we considered the animals to be resolved. And what you see here is female mice resolve their infection spontaneously over time, whereas male animals stay infected. And what I'm showing you here is where about 50% of our male animals stay infected, but we have experiments where up to 100% of the males are infected for the entire length of the experiment. So if we look a little bit more closely, what we see is that at that initial time point, the number of bacteria that are in the bladder that are part of the infection are actually equal between female and male mice. So what we've done here is we've digested the bladders from these mice that are infected, and we count the number of bacteria that are present. Each dot is a mouse, and again, red is female and blue is male. And what you can see is the number of bacteria declines steadily over time in female mice until they reach this sort of reservoir a concentration that we know exists in the tissue. But in male mice, what we see is they remain chronically infected up to 1 month post-infection, and they have a significantly higher bacterial burden 28 days after infection. So to get at why this might be, we looked at the very early immune responses, and we know in female mice, the cytokine response peaks at about 24 hours post-infection. So what we did first is we took these same bladders that we ground up to assess how many bacteria were present, and we also asked what cytokines are present. So what you're looking at here is called a radar plot, and so these are all of the different cytokines that are significantly differently expressed between a naive female and an infected female. And so you can read it by reading the cytokines on the outside, and what you see here is a log scale. So in red are 24-hour infected female mice, so you can see that they express very high levels of cytokines. And then in blue are the male mice, and you can see for many of the cytokines that we measured, they express lower levels, and in some cases significantly lower levels of these cytokines compared to female mice. Then the dotted lines are essentially animals that were treated with PBS, and that's just to control for any damage that might occur when we catheterize these animals. And so we thought, okay, the response in female mice is clearly stronger at 24 hours, but maybe it's just delayed and it comes a little bit later in male mice. So we looked again at 48 hours. And what we saw is that not only was there not a delayed response in male mice, but in fact, the cytokine response in general in female and male mice collapsed down to that essentially similar to what we see in completely naive animals. And so what that says to us is that the cytokine response, or in general, the immune response to this infection, is very early, it's very acute, it happens in the first 24 hours. And so at this point, we were a little bit concerned that what we were looking at could potentially be just a mouse phenomenon, even though it essentially recapitulates what we see in the clinic, which is women have more infections, but whenever a man has an infection, it's considered to be a complicated infection. It lasts longer, it has a greater tendency to go chronic, and it's harder to treat with antibiotics. So what we did is we collaborated with a good friend at Institut Pasteur, who's co-leading the milieu interior cohort. So essentially this is a cohort of 1,000 healthy individuals aged 20 to 69 who have donated a number of different samples, including blood. And what we had is the samples that had donated, blood samples they had donated and then had been subsequently stimulated with an E. coli stimulus. And so what we looked at were a nanostring analysis of about 560 different immune genes. And so what I'm showing you here is the result of this analysis. So this is expression of 560 genes in female or male donors between the ages of 20 and 50. And we focus on those younger donors because we're really curious about this time point in the lifetime where the infection is is the greatest. And so what you can see here is if we overlay the sex of the donors on this gene expression, we can see that female and male donors actually pull apart, and that the female donors cluster together and the male donors cluster together. And then what we did is we looked to see the different cytokines that we know are differentially expressed in the mouse. We looked to see which ones we had in that nanostring analysis, and we overlaid their expression on this PCA plot. And so what you can see here in red now, this is higher expression, and green is lower expression. So what you can appreciate from these 4 cytokines that I'm showing you here is that that higher expression of CXCL2, IL-8, LIF, and IL-1 alpha are all higher in the female donors compared to the male donors. And so this really mirrored what we were seeing in our animal studies and gave us a little bit of encouragement to move on. And so, of course, if you have different cytokine expression, you can expect that this will impact immune cell infiltration. So the next thing that we looked at is immune cell infiltration into the bladders of infected animals. And what I'm showing you here now is 0, 24 hours, and 48 hours post-infection, and this is always on a log scale. So we see at baseline uninfected animals, the number of total immune cells is actually the same between female and mice. And so the difference in the immune response is not based on something that's already sort of naturally there. But what we do see is that at 24 hours post-infection, and this is reflecting these differences in cytokines, we see an increase in the total number of immune cells that infiltrate the bladder in female mice compared to male mice. And we see at 48 hours, those differences essentially disappear. We don't really see any differences in resident macrophages, but we don't actually anticipate that either because it's a resident cell. They don't infiltrate. We see a very small difference in the number of dendritic cells that are there. But what we really see that was quite striking is a massive difference in the number of neutrophils, monocytes, and eosinophils that infiltrate the bladder 24 hours after infection. And this is much higher in female mice compared to male mice. And you can see overall that we have an increase of 100 to 1,000 times the number of cells that are resident in an uninfected bladder. So we were thinking quite a bit about this, and what really was sort of important and what we appreciate is that hormone changes mark these different stages of life. And so I've just put those same little incidence dot plots down here in correlating with pre-adolescent and adolescent and adults and individuals who are over the age of 50. And so we thought, okay, maybe the sex bias that we're seeing in urinary tract infection is actually due to a differential immune response, which is influenced or regulated by sex hormones. So to test this hypothesis, we took a relatively simple approach, and we thought we would just begin by removing testosterone from the system or adding it back in. And so I'm just showing you the controls here. If you measure testosterone levels, you can see that there is testosterone present in female mice, but that it's really quite low. We see that there's variable levels of testosterone in male mice. This is reported. This is common for anyone who's worked with animals, and you see that one animal has groomed the rest of them. That's an animal that has the highest level of testosterone in the cage. And what we did is we castrated male mice, and what you can see now is we can reduce the level of testosterone to that, kind of similar to female mice. And in female mice, what we did is we used something called a T-tube. And so that's a tube where you can put testosterone in, and then you implant it under the skin of the animals, and it slowly leaches out over time. And what you can see here is we can raise the testosterone level in these animals to high but physiological levels of testosterone. And so this is what we did. So in the animals that had been castrated, what we saw is this actually did not impact resolution of infection. So these animals stayed infected for the duration, and they had similar bacterial levels in their bladders at the end of the experiment 28 days later. In contrast to this, what we saw in animals that had been treated with testosterone is female mice were now no longer able to resolve their infection. So they stayed infected for the duration of the experiment, much like male animals, and they had higher bacterial burdens compared to the untreated animals. And so this suggests to us sort of an acute action of testosterone in modulating how the animals can resolve, but also probably sort of an imprinting as well on the animals, that even though we remove the testosterone, we still see that there are effects present in these mice. So in trying to understand what was essentially mediating this inability to resolve, we looked at, again, the cytokine expression in animals that had been treated with mock tubes or had been treated with testosterone. And we saw that a subset of cytokines were reduced similar to what we saw in male animals, but one thing we didn't see is myeloid cell infiltration was absolutely unaltered. So that couldn't explain why the animals couldn't resolve their infection any longer. So we thought, okay, what's going on here? We have this first scenario where we have male mice that cannot clear their infection compared to female mice. We have animals, female mice that have been treated with testosterone who now are no longer able to resolve their infection. And we had, from a completely unrelated experiment in the lab, this scenario where rag gamma C animals could not resolve their infection. So rag gamma C animals are animals that are missing the gene to recombine, so they don't have B cells and T cells, and they're also missing the common gamma chain, which means that they don't have NK cells, gamma delta T cells, innate lymphoid cells, a number of different immune cells, and they are also missing cytokine responses. So while this is sort of a profoundly immune deficient animal, it gave us some clues about what cells we wanted to actually look at. And so what we did is we went back and we looked at lymphocytes and their infiltration into infection. So what you're seeing now, again, are naive female and male animals and infected 24 hours next to them. And what we saw is an increase in CD4 positive T cells, NK cells, gamma delta T cells, and this really specific CD4 positive innate lymphoid cell, group 3. And what you can see here is we saw these increases only in female animals over time, and only gamma delta cells are actually different between naive male and female mice. We then went on and we did sort of the opposite experiment, and we looked in animals that had been treated with testosterone, and we saw that these same populations were decreased in the animals that had been treated with testosterone compared to their untreated controls at 24 hours post-infection. And so what's really interesting about these 4 groups of cells is 3 of them make the cytokine IL-17. So if you were paying attention, or if you go back and watch the recording, what you'll see is that IL-17 is one of the most differentially expressed cytokines between male and female mice at 24 hours. And that difference actually persists at 48 hours, and it's one of the only cytokines that's still different at 48 hours. And so we thought, okay, all arrows point to IL-17. And so what we did is we neutralized this cytokine at the beginning of the infection in female mice. And what we saw is that animals that had been neutralized for the cytokine now were no longer able to resolve their infection compared to the mock treated or isotype treated animals. We subsequently went on and we used a delta TCR mouse. So this is a mouse that's missing gamma delta T cells, which are a major source of IL-17. And we see an intermediate phenotype here. And so that suggests to us that gamma delta T cells are not the only cell that's providing IL-17, but they probably are providing some sort of a major portion of that cytokine that we need for resolution. And so if I kind of sum up what we understand from our infections is we know that we infect these animals and they're recognized. We know they're recognized because we see a burst of cytokine expression, including this damaged cytokine IL-33. And essentially what happens is this cytokine will recruit, this whole entire cytokine milieu, if you will recruit neutrophils and monocytes to the site of infection. Those monocytes will come in, they'll differentiate into macrophages, and there's also resident macrophages that partake in the immune response. And ultimately, we know that we get this really diverse mix of effector cells that seem to be able to take care of the infection. Now, if we look at the male mouse, we see really a completely different scenario. What we see is even though we have an infection, we just don't have that recognition. We don't have the broad cytokine response, and the result of that is we don't get infiltration, and ultimately we don't get those cells that come in that help mediate resolution of infection, and this leads to chronic UTI, which we can see here. So from this, where are we going now? What are the questions we know? So basically what we know for sure is that this decision on sort of whether you will be able to resolve your infection or if you're going to go chronic happens in the first 24 hours. And so that made us think, let's look at some of the earlier or sort of initial interactions. And we asked the question, are these first interactions in the animals different? So if you remember, I told you at 24 hours, the number of bacteria present in female mice and male mice is actually the same. So we looked much earlier. We looked at 1 hour post-infection. And what we saw is that female mice had a significantly higher number of bacteria in their bladder compared to male mice. And the consequence of this is we actually saw a differential NF-kappa B activation. So in female mice that were infected, we could see that NF-kappa B was activated, it translocated to the nucleus, and we just didn't see this in male mice. And that led us to question whether the interactions of the bacteria with the bladder surface could potentially be different. And so Laura Ramirez-Finn, who is a PhD student in the lab, now looked at this during her master's work in our lab. And what she did is she used scanning electron microscopy essentially to look at how the bacteria are interacting with the surface of the bladder. And while these are preliminary images, and I think the best part about scanning EM is you can speculate madly, what we were able to see is it looks like that the bacteria may be interacting with the surface of female bladders in a different way from male bladders. And so when we look, we see these bacteria that are oriented perhaps as if they're diving into the surface of bladder, and we see these what look almost like craters with bacteria inside of them. Whereas when we look at the male mice, what we see is the bacteria appear to be more lying on the surface of the bladder, so they're more horizontal, and you can see one lying right here. And so this is something that we're trying to understand right now. And ultimately what we have is a model of the very first interactions, including NF-kappa V activation and cytokine expression leading to cell death and the inflammatory response. And so what we're doing is we're looking at each of these early steps to see where there are differences in male versus female mice to understand essentially what initiates this difference in the immune response and what amplifies it. So in addition to that, we're actually also trying to continue our comparison with our animal studies in humans. And so to do this, we've gone back to working with the milieu interior again, and this is the work of Leah Delturbe, who's also a PhD student in the lab. And what Leah is doing is she's taking these gene expression patterns we have, so all of this nanostring data, and she's essentially asking a question: what genes are differentially expressed between females and males, women and men, in different conditions over time? And so what I'm showing you here is just really just the tip of the iceberg of her analyses. But what she did is she looked in this homeostasis condition. So these are individuals who have not been stimulated with any like E. coli or any other bacteria, any conditions like that. And what she's able to see is that there are genes that are differentially expressed between young donors who are female versus male. And those genes, in fact, the number of them decrease when we compare male and female donors between the ages of 60 and 69. And then when we look in the E. coli stimulus condition, what she sees is, again, there are genes that are differentially expressed. The number of them diminish over time when our donors age. But in addition to there being differences in the number of genes, the actual genes themselves are different between the homeostasis condition and the E. coli condition. And essentially what she's doing is she's working under the hypothesis that genes that are differentially expressed between the sexes only in young donors may be due to differences in sex hormone levels, whereas genes that are differentially expressed across ages could potentially be due to the effect of sex chromosomes. And ultimately, she'll take these analyses and correlate them with sex hormone expression in each of our donors. So I like to say this is the point of a shameless self-promotion. This is an article that we recently published. So if you're interested in learning a little bit more about biological sex and its impact on diseases of the urinary tract, I invite you to check this paper out. And with that, I'd really like just to say thank you to really my excellent team. This is when we were at the Institut Pasteur. And this is Mathieu Rousseau and Livia Lacerda-Mariano, who did a lot of those infection studies I showed you. And this is the team now at Institut Cochin. And here's Leah right here, and here's Laura, who are working on the sex differences in humans and mice, respectively. And I'd just like to acknowledge our funding and all of the excellent support that we get from the core facilities at our institute. And with that, I'd be really happy to. No, it's not up to me yet. It's up to Neeloffer to lead us into our discussion. So thank you very much.
Thank you so much for that excellent talk, Dr. Ingersoll. We'll now start our Q&A session. And just to remind the audience, please type your questions in the question module in the chat box there, and I will monitor the questions. So the first question, and this is, let's start with a question for Sabra. I'm interested in the gender gap in antibiotic prescriptions, where women are more likely to be prescribed antibiotics than men. Is this clinically appropriate? I understand it is often explained by consultation behavior, and interested in your opinion. This comes from Will Robertson.
Well, thank you so much. Such an important question. So my understanding of the literature is consistent with yours. So women are being prescribed antibiotics more often, and I think the literature suggests that this is a gender bias, probably having to do with over prescription or even misdiagnosis in women, because I can tell you as a viral immunologist, there is also a body of literature showing that women are more likely to be incorrectly prescribed antibiotics for viral infections. And you know, maybe kind of misdiagnosing and overdiagnosing women. This goes beyond infectious diseases. There's a very rich body of literature for heart disease showing how women presenting with symptoms that can be somewhat different from men can contribute. And I think this has been grossly understudied in infectious diseases. I think there is an assumption that we are all going to present and in the same way. And, you know, I think long COVID is starting to raise these interesting questions that we may not all always present in the same way. So thank you.
Thank you so much. And I apologize for mentioning the name of the person who asked the question. I just got a note from our organizer that we are not supposed to mention the name of the person. So the next question is for Dr. Ingersoll. And the question is regarding the intersection of sex and age. How can sex age impact differences be managed during preclinical animal studies and in clinical trials of new treatment for infections?
And maybe we both can take this, because I think that Sabra made a really great point when she was talking about essentially estrogen replacement. It's funny, I had to laugh when you said maybe it's your age, but you're thinking about it, because it's on my mind too. And there's some really great studies that show that this actually alters your immune response and potentially elevates it back to your premenopausal. So I mean, that's certainly an option for women, right? And one of the things that we think about when we talk about this impact of what we're seeing with testosterone, for example, in the response to urinary tract infection, there are treatments. There's androgen deprivation therapy, and that's used frequently for prostate cancer, for example, right? And we can imagine that this could be a possible sort of short-term solution when we have an individual who is facing chronic infection. And so I think what we want to do is open up our minds to considering sex hormone levels at potentially at all ages, because there's certainly a lot of variability in younger individuals as well. So open up our minds to considering that when we're thinking about treatment protocols. And I think if we do that, we can essentially incorporate that into treatment and maybe have better responses.
And so related,
I love that. You know, and the only thing I would love to add to what Molly just beautifully described is, you know, we saw that to some degree. There were some beautiful data during really the worst of the pandemic in Italy, showing that men with prostate cancer who were on androgen receptor antagonists seem to be doing better, seem to be less likely to be hospitalized with SARS-CoV-2. So I do think these notions about how we might use sex steroids therapeutically, I think that door is starting to be opened.
Yeah, I agree.
So related question here for Sabra, when a vaccine response is estradiol dependent in older females, for humans, is it feasible to give estradiol with the vaccine?
Oh, that is such a good question, and something that we have been asked. I think it is feasible. I think, you know, we haven't actually tested it with just a single dose of estradiol to see if we could drive a broader, kind of a greater response. And I think that is wonderful. And that is something that we could incorporate into experimental design in our preclinical animal studies to see if all you need is, you know, kind of a bolus of the hormone at the time of antigen exposure versus staying on these treatments for longer term, which is what we did in our study. So I love the question, and I don't have an answer, but something I think we could readily address.
So there are two questions and they're related, so I'll summarize this. And these are to both speakers, essentially asking the significance of the role of sex steroid receptors or hormone receptors and the response elements in the genome to explain biological sex difference in infections. So Sabra, you want to go first and then Molly?
So that sounds great. So such a good question. I think a lot of the way by which sex steroids have their effects are through these classic binding to an intracellular receptor, and then that receptor hormone complex translocating to the nucleus and binding to a hormone response element in the promoter of a gene and transcriptionally regulating that gene. That's not the only way. And, you know, I can give other examples. You know, I love that Molly spoke about NF-kappa B. You know, it does have, for example, an estrogen response element, and we do know that it can be transcriptionally regulated, and that many other genes, including TNF-alpha and IL-6, do not necessarily need to be regulated by estrogen through an estrogen response element, but it's more through an effect on NF-kappa B. So I think you can appreciate the complexity that there can be diverse kind of direct as well as indirect ways by which these steroids can regulate some of the responses that we've described. I'll leave it to Molly to add.
Yeah, I cannot underscore how important it is to appreciate the complexity in this. It's we dove in very naively, and now we're facing a lot of these questions, and we're like, but this is actually one of the reasons why we pulled in analysis of sex chromosomes, right? Because we know that there's, you know, more than 1,000 immune genes expressed on the X chromosome. There's about 100 expressed on the Y chromosome, and we know that sometimes we can look at sex steroid receptors and we don't see any differences or we don't see any changes, but once we have a inflamed microenvironment, this changes their expression, and a lot of times it upregulates them. And so it looks like they're not peripheral, they're really key players in cytokine expression. They're present in all sorts of different immune cells to different degrees, and we know that essentially you need to be sort of monitoring them through the course of disease, because they will change before, during, after, perhaps during the acute, perhaps during the chronic stage of infection of any type. So.
Yeah, and it's definitely, I mean, this is a question that I would like to ask both speakers very briefly, if you can give us some ideas as to how can one, perhaps an animals model, really delve into the question of teasing out what response is dependent on sex steroids versus chromosomes.
That one is easy. Can I say that? Certainly in animal models, there's a model that we're trying to get into the lab right now. So this is the 4-core genome mouse. And so this is an incredible model. Essentially what you have is 4 different animals. So if you understand that the SRY gene, which is responsible for testes development, is typically on the Y, well, on the Y. So what they've done in this model is they've taken that gene, they've moved it to chromosome 3. And so in how you cross the animals, what you get is an animal that can be XX or XY, but be female, or XX or XY and be male, as long as they have that SRY gene on their chromosome 3. And so then what you can do now is you can ask questions about gene dosage. So you can have a female animal that has 1 or 2 X's, or a male animal that has 1 or 2 X's. And through gonadectomy, you can then remove hormones, and so you can ask just chromosomes. And this model was actually used to show that there is a gene that's protective on the X chromosome and is protective for the development of bladder cancer and seems only to function in female animals and potentially in women as well. And so it's a really exciting tool that I think is potentially the most straightforward way to ask those types of questions.
And the only thing that I would add is, you know, we have these opportunities in humans through individuals who have genetic modifications. So individual, you know, females who have Turner syndrome and have one X chromosome. And then we have Klinefelter men who have 2 X chromosomes and a Y, so you have some of the differences in the X chromosome dosage, as well as the presence of Y in Klinefelter syndrome. And I think, you know, I think there's growing interest in better understanding in transgender people. Do they start to show a disease susceptibility profile that resembles their transgender after receipt of gender affirming surgery and hormone replacement, or do they still maintain from their birth sex certain susceptibilities? And we're starting to see this. I haven't seen this as much in the infectious disease realm, which is our focus today, but I am starting to see this and beautiful publications in the context of autoimmune diseases, where, you know, you have a staggering statistic in which roughly 80% of all autoimmune disease patients are women. And I think that was shown and mentioned in our introductory remarks.
Yeah, thank you so much. And so the next question here for Molly, given you see difference in adhesion to bladder epithelial cells due to sex, what is your understanding for the difference once the pathogen invades into the host cell? Do you think sex plays a role in relapse of infections and formations of QIRs?
Okay, I can answer part of that for sure. David Hunsted's labs actually published that there aren't really any differences. Well, no, he looked more at the acute communities that form. He did not report any differences, and we confirmed that. We didn't publish it, but we confirmed we don't see differences in the numbers of bacteria once they've invaded and how they develop into these communities. In terms of QIRs, we've done it more, we've addressed it more using counting CFU rather than using imaging to look to see where the bacteria are. But we see that these reservoirs that form, we get equal numbers in male and female mice. The caveat being that we absolutely have to treat the male mice with antibiotics before we can assess this. So we need, that chronic infection needs to be dealt with first. And we've done the control of treating female mice with the antibiotic at the same time, just to make sure we're not altering anything. And so in terms of that, we think that's pretty much the same. But those initial interactions, I think the question we're really trying to get at is whether the sex bias that we see is exactly in how the bacteria interact with the surface, how they invade, or how they're sensed once they're inside of the cell. And I think when we can get at that, then we'll be able to understand what's sort of amplifying that whole signal. But once they're in, really by 24 hours, it looks like everything's sort of equalized, at least for the bacteria.
So this is really cool. And this question to both our speakers, and a couple of individuals have asked this question, and I think I'll summarize this, is there are some cytokines you show, both of you have looked at inflammatory cytokines, and some cytokines you show have a clear sex difference in a short period of time, which then is mitigated in a longer time point based on the kinetics you're looking at. Excellent example, Molly, you gave was IL-17. So we will first go to Sabra and then Molly. Comment, if you could comment on, are there some specific cytokine responses or inflammatory responses that you think is independent of the action of sex hormones or sex steroid receptors, and more chromosomally influenced? Sabra first and then Molly.
That is a wonderful question. Absolutely, there very likely are, in part, as Molly noted, in that, over 1,000 genes that are encoded on the X chromosome. There are several immune response genes that are present on the X chromosome. And in particular, there are some receptors. One that I'm thinking about off the top of my head is IL-13, which has been shown using that 4-core genotype mouse model that Molly elegantly described, and showing its important role in modulating sex chromosomal effects in autoimmune disease models, specifically a mouse model of multiple sclerosis. So I do think there are, and we will continue to uncover some of these. I do think, though, it's going to probably, biology is sometimes beautifully complex, which can be very frustrating from an experimental perspective. And what we're noting, even with our toll-like receptor 7, which is X-linked, it is responsive to estrogen as well. So just because something is X-linked doesn't mean hormones can't impact it. And I'll turn it over to Molly.
Yeah, I think that's exactly it. I think that's what we're kind of up against, is we have this hypothesis that it's hormones or chromosomes, and I think Leia's listening. I think that's ultimately wrong, because I do think that they're going to intersect. I do think they're going to interact. I wanted to ask Sabra, you must come up against IL-17 as well, because there's some really nice publications showing IL-17 is different in the lung as well, right?
There absolutely are. We have yet to see this in our flu model, and we really haven't explored this in our SARS-CoV-2. So that's to be determined. But in our influenza model, we really have yet to find IL-17 either dramatically different between the sexes to really explain some of the sex differences, or dramatically changed in during pregnancy, either in the lungs or in the placenta, where we do look at several immune responses. So, but your point is so well taken, and something that we will be exploring in our SARS-CoV-2 mouse model.
I'm curious. Look out for it.
Yeah, I will let you know.
No, it's completely astonishing how IL-17 pops up, the sex difference in IL-17, much more pronounced in females compared to males. For example, we study allergen-induced asthma, and there you go. IL-17 was one of the most sex-biased cytokine in the lungs that we showed. So IL-17 definitely pops up, and we'll keep an eye on the story as we move forward as a community. So here is another IL-17 question for you, Dr. Ingersoll. Let me summarize this. Whether estrogen was introduced similar to the testosterone experiments that you showed. So did you do exogenous administration of estrogen and see any change in infection resolution state? And the second part to this is that as IL-17 was significantly low in males during the infection, does this change when you introduce either estradiol or testosterone?
Right. So we did do the estrogen experiments. We did add some estrogen back in, and we don't see a change in resolution of infection. In fact, what we do see are changes in the number of circulating immune cells in male mice, but not in female mice. And it does look like we can elevate, estrogen does help elevate the number of immune cells that infiltrate during infection, only in male mice, not in female mice. And so that's something we're working on right now to understand that. And what's a little bit curious is we can bring those numbers up, but it still isn't sufficient to change resolution. So there's something else that's going on at the same time. So we can get that response up. And mostly we focus on those myeloid cells, and now we suspect that the myeloid cells are not the only cells that are responsible. So we need to go back and understand if those IL-17 producing cells specifically are impacting this. We haven't looked at cytokine expression in these animals yet. So that I think would be an interesting thing to look at. One thing we did do though is we did supplement male mice with IL-17, and that was not sufficient to mediate resolution. And so our idea there is that IL-17 is really different, but it's accompanied by 24 other cytokines that are differentially expressed. And so what we think is it's necessary, but absolutely not sufficient to restore that entire inflammatory milieu we need for resolution. So I think I think it has friends. It's working with something else there that we have to sort out.
Yeah, absolutely. Sabra, I mean, you work with autoimmune disease as well as infectious disease. So just curious, in terms of sex-related differences that you see in your respiratory infection models, do you think there's some intersectionality between autoimmunity and susceptibility to these infections and the sex bias in that?
I absolutely think there is, and I'm going to give you, I'll give you one example, and then I'll expand what you're asking. So one great example is toll-like receptor 7. So while in the context of vaccination, I'm illustrating that it's beneficial that driving antibodies to make, or driving B cells to make antibody to a vaccine antigen is a good thing. And in our challenge studies, it results in greater protection from infection. And I didn't show you that if we knock out toll-like receptor 7, we lose that female advantage. So we know it's intimately involved. Now let's talk about lupus. 90% of lupus patients are female. Take that same TLR7, it is more highly expressed in B cells from female lupus patients than even male lupus patients. And this is driving greater antibody production against self-recognized, in this case, DNA. So there it is highly detrimental to have this same biological difference. And the way I want to expand this is, you know, in your introductory remarks, you really showed how autoimmune disease and non-reproductive cancers are kind of at the poles. And what I'm starting to see in the literature and in my review of the literature of those areas is that some of the very same cellular activities that contribute to autoimmune disease in females make females less susceptible to developing some of these cancers and vice versa. So, you know, and now we're starting to see increased interest in, you know, and we're seeing sex differences in how well things like checkpoint inhibitors, so immunomodulatory therapies work. So I think really where I'm seeing a real explosion and where I think we're really going to start to see sex modifying factors being considered in treatment is going to be in cancer. And I think the rest of us are going to benefit from that because it's really, I just see such growth in that area. And I don't even work in that area, but I feel like all I do is review papers in that area, and it's really exploding.
No, I mean, that's excellent. And there are very few researchers who study these models of autoimmunity and infections in parallel. And so the synergy of the two, or the intersectionality of the two, which is clinically so important in preclinical studies, is often kind of neglected.
I completely agree with you. And we even see that for many autoimmune diseases, you know, multiple sclerosis would be a great example. For reasons that are not still completely understood, there's greater susceptibility, not just because of the immunomodulatory medications that people are on, but there's likely something biologically going on that increases susceptibility to certain infections, including respiratory infections.
Right. Yeah, great discussion. We can have a complete webinar on this. So the next question for you, Molly, can you expand on what you think might be the differences observed in the position of bacteria in the bladder of mice on EM. Immune evasion. Is it immune evasion?
Oh, okay. Yeah, I know it's getting really dark here. I think I'm getting close to the end. So here's my favorite hypothesis, and we will test this, and we will know in just a few short years. But my favorite hypothesis is that the mucus itself is actually different between the male and female mice. And so there's a little bit of precedence for this in other mucosal tissues in general, but there's absolutely nothing known about the mucus in the bladder. But we know in general things like estrogen can impact mucus thickness, for example, and we see that in the vaginal tract. So we're working on the hypothesis that there are differences, and these differences in the mucus in a homeostatic state is what is impacting how the bacteria are interacting with the surface of the bladder. So really, this is a really exciting, brand new project, but we've kicked it off pretty strongly with all the omics we can possibly think of to look at it. So.
So do you feel, based on your experimental evidence in UTI, that men and women perhaps need differences in the duration of antibiotic treatment?
I think yes. And I think that some of that is because we know, and we know from our animal models, and I suspect that the human clinical data supports this, but in women and female animals, the bacteria are in the bladder. Sometimes they go to the kidney. In male animals for sure, and likely in humans, the bacteria are in the bladder, they're in the prostate, and in the mouse, they go to the seminal vesicles, the testicles, the kidney. So they're everywhere, right? So we have multiple infections happening at the same time. So that means we probably need more antibiotics. And we know that there's a difference in penetrance in how those antibiotics reach those different tissues. And I wasn't going to talk about it, but it's just super exciting. We have very preliminary data that suggests that testosterone may impact the ability of certain antibiotics to kill bacteria in vivo. So this is something we're working on right now. And again, this comes back to that, that idea that maybe we need to modulate the levels of sex hormones in the context of treatment for infectious disease and potentially for other diseases as well. I think Sabra's point about cancer and everything is absolutely fantastic. So, maybe there are real biological reasons why we can't treat the exact same infection in the same way between women and men.
So in closing to our Q&A session, because we have a little bit of time, I was wondering if both our speakers could give some closing remarks. It's completely up to you on sex difference in biomedical research or infectious disease. So I'll start with Dr. Klein and then Dr. Ingersoll.
So I think what we have both shown and what the literature really suggests to us is sex is providing us with tremendous insights about therapeutic modifications that could be made to improve outcomes. And I do think we have to be prepared that some of these might be sex specific. And as we move toward personalized medicine, it may not be that each of us always has to be treated differently, but yet thinking about the sex, and I think what we showed, thinking about our age, how our hormonal changes with age contribute to this, we might find that we're going to be treated differently.
Molly?
Yes, that was excellent. I completely agree. I think the only thing I would want to add is we all, as a community of researchers, need to consider sex and potentially gender when it impacts what we're doing in all of our studies. I appreciate that doubles your research budget, but we have to be prepared to be completely open-minded and to look at the impact of biological sex in ways that we never thought that it might play a role. So I encourage everyone, add it into your budgets, add it into your grants, and please consider sex differences as a potential contributor to whatever system that you're looking at.
Thank you to both our speakers. That was an excellent webinar, and now it's over to you, Astrid.
All right. Yeah, thank you. I agree, Neeloffer. It was really a really exciting webinar, and I'm very thankful to both our speakers for your really very specifically engaging presentations. And of course, Neeloffer, thank you for doing a great job with the moderation. And of course, I also will thank the audience for their attention. But before I let you all go, I need to tell you about the upcoming webinar. So in January, we will have a webinar about delivery systems in antibacterial drug discovery. That will be on the 24th of January. It's not yet on our website, but it will be added there soon, a registration link. And of course, also everybody who registered for today's webinar will receive a follow-up email in the coming weeks with the registration information for the next webinar. And yeah, with this, I would like to thank everybody for the attention and for the contributions. And as always, I'm asking you, please share our information, share the webinars, share the content widely, and yeah, join again. Thank you and goodbye.
Thank you. Bye, everybody.
Bye.